All right, so what we're going to look at today, remember I said yesterday that we're going to look at DNA technology. Devin and Gowan. So we're going to look at DNA technology, but in order to understand some of the things that we're going to use with DNA technology, there's some things about bacteria and viruses that we have to understand. All right, so we're going to look at viruses today and wrap that up tomorrow and look at bacteria. Bacteria is a little bit shorter in terms of that. So this is a E. coli bacteria in the large, with the viruses, the bacteriophages that are infecting that. So bacteria and viruses have been studied immensely, basically because a lot of them can cause disease. They're pathogenic, and so we have interest in them. Bacteria cause about half of all human diseases, and they typically cause disease by producing either exo or endotoxins. So exotoxins are secreted and cause disease even if the bacteria that produce them are not present. And so, therefore, sometimes the bacteria produces a toxin and then you come into contact with that toxin and can get sick and not necessarily be infected with the bacteria. And then endotoxins are released only when bacteria die and their cell walls break down. And so then that comes from within, and so you have to be infected with the bacteria for this. So oftentimes our adverse reactions and sickness can be caused by things that the bacteria produces and that are affecting our reaction against them. So an example here, Lyme disease. I'm sure you've heard of Lyme disease. It's caused by a bacteria. No? No? Okay. All right. Lyme disease is caused by a bacteria that is transferred from one organism to another via a tick. So the bacteria is carried by ticks, and so what happens is people can get infected with Lyme disease by being bitten by a tick. And then, so then what happens is that bacteria produces, instead of toxins, they produce proteins that are released and compromise the host's immune system and can cause Lyme disease. And so on the left here is the tick, and this is a rash that is typical, what typically looks like when you've been infected with that particular bacteria. Lots of different organisms, bacteria can be spread by ticks. There are certain ones that can be spread by bacteria or by mosquitoes and things like that. And so Lyme disease is one of those. So since we're talking about bacteria and viruses, I put this picture up when we talked about cells, but I just want to show you size-wise. So this is just a small portion right here of an animal cell, and this is an animal cell nucleus. And this is a bacteria, so bacteria is much smaller than the animal cell. Virus is way, way smaller than a bacteria. And so in just a minute we're going to write, I have in there that the smallest virus actually is much smaller than a ribosome in an animal cell. So they're really, really, really small, these viruses, comparative to other cells. All right, so let's look at viruses. So viruses are basically a genome, that's their genetic material, enclosed in a protective protein coat. So we looked at viruses that infect bacteria. So they have just a protein with the DNA inside of them. And so the genome is the genetic material. Viruses typically are not considered alive. Well, some people make arguments against that. Can somebody tell me why somebody might not think that the viruses are alive? Yeah, they cannot reproduce on their own. So if their main components are a genetic material, let's say it's DNA and a protein coat. So what the virus has to do in order to reproduce is it has to replicate its DNA, and it has to make new proteins. And the virus themselves don't have the machinery to do that. So they don't have the enzymes to do DNA replication. And therefore protein synthesis as well. And so what they do is they basically inject their genetic material into a host cell, and then use the host cell's machinery, all the enzymes and so on, and let the host cell copy its DNA and make new viral proteins, and therefore let the host cell make viruses, and then those viruses can go infect other cells.
So when they say like kills like 99% of viruses, is that just like deactivating them?
Yeah, so it makes it not so they can't infect you. So the killing part is like destroying them. Yes, absolutely. All right. Tiny viruses, this is what I just said, are only 20 nanometers in diameter, smaller than a ribosome. So I've already talked about that. So depending upon the type of virus, one way to classify viruses is their genome. And their genome is their genetic material. All of our cells and cells of eukaryotic organisms and bacteria, all of our genetic material is DNA. Viruses are unique in that some viruses, their genetic material is RNA. Some viruses have no DNA inside of them. Their genetic material is RNA. So depending upon the type of virus their genome consists of, it could be double-stranded or single-stranded DNA, or double-stranded or single-stranded RNA. So unlike us, we have in our cells DNA. All of our DNA is double-stranded. We have two sides. And then we can use that DNA to make RNA, and all of our RNA is single-stranded. Well, you can see here that bacteria, if they have DNA as their genetic, or viruses, if they have DNA as their genetic material, yes, it can be double-stranded like us, or it can just have one side of it and be single-stranded. And then RNA, it can be single-stranded like our cells, or double-stranded. And so that's one way to classify viruses. Then this DNA or RNA is surrounded by the technical name of the protein code is called a capsid. So it's the shell enclosing the virus's genome. Some viruses have something beyond the capsid. They also have structures to help them to infect their host cells. For example, a membrane envelope. We're going to see in just a little bit here that some viruses have this envelope that surrounds the capsids, and flu viruses are like this. And so if we look at this picture here, this compares and contrasts some different structures of different viruses. On the bottom here, it tells you what type of virus it is. This is an actual electron micrograph picture of the virus's computer-generated picture to show you the parts. So the bacteriophage, we've talked about this before. So in purple in all of these pictures is the capsid, the protein code. And then the genetic material is inside each one of these capsids here. So a bacteriophage has a protein coat with the DNA inside of it. The flu virus is a little bit different. It has capsids inside, and each capsid, each protein coat surrounds its genetic material, but it's all surrounded by a membrane. And so the flu virus, we're going to see here that this membrane around it helps the flu virus infect our cells, and we'll look at how that does. So it helps to infect certain cells. And then these guys are like this one here, that they don't have a membrane, but you can see the different shapes and so on. So, but they have the capsid surrounded by the genetic material. So lots of different viral structures. Each virus is a little bit different. But what they all have in common is that they need a host cell. So they reproduce only within a host cell. Each type can infect a limited range of host cells. So what does that mean? So different viruses infect different types of cells. And sometimes it can infect multiple types of cells. Sometimes it's very specific. Sometimes it's very specific, like a virus will only infect one type of species. Sometimes that virus, when it infects the species, only infects certain cells within that species. So like for instance, the cold virus only infects the cells of our upper respiratory tract, and so it doesn't infect the cells of your skin, and so on. And so sometimes they can infect a limited range of different species, so they can infect humans and other species as well, so therefore can be infect and go across species. So that's what it means by limited range of host cells. Why can it do just a limited range of host cells? It has to do with the fit of the proteins on the virus surface and specific receptor molecules on the host surface. So what happens is if you have, this is a cell, and this is a virus, what makes this virus be able to infect this cell is that the proteins surrounding this, on the surface of this virus, fit on the receptor proteins on this cell. And if this virus matches, then it can inject its genetic material and use it to make new viruses. If the cell doesn't have the right receptor, it can come into that contact with that virus, but that virus is not going to affect it because it doesn't have the right receptors on there. And so that's why, maybe another animal can get infected with a virus, and you can come into contact with that virus, but if it doesn't have the right receptors, it's not going to affect you. All right. So some viruses can infect several species. That's what I said here. While others infect only a single species. So examples, just to give you some examples, West Nile virus can infect mosquitoes, birds, horses, and humans. So it's a cross species there. Measles viruses only affect humans. Most viruses that infect eukaryotic cells, like our cells, do attack specific tissues, and I put here that, as I just mentioned, the human cold virus infects only the cells of the upper respiratory tract, so it's very specific in the types of cells. The AIDS virus binds only to certain white blood cells. Put in parentheses here, AIDS virus, that's the HIV virus. So AIDS is the disease caused by the HIV virus. Okay, so how does a virus infect a host cell? So the viral infection begins when the genome, either it's a DNA or RNA virus, enters the host cell. So the genetic material enters the host cell. Once inside, the viral genome takes over its host, causing the cell to copy viral nucleic acid and manufacture viral proteins. So viral nucleic acid, so that's going to be either DNA or RNA, depending upon, it's supposed to be an R, depending upon the type of virus it is, if its genetic material is DNA or RNA. So it's going to use the host cell to make new viruses. And remember, the two main components of the virus is the genetic material and the protein. So it's going to use the cell to make both of those. And so what it does, the host cell that is providing the materials needed. So what are those materials? Nucleotides, amino acids, energy for ATP to do this, for making the viral components, dictated by the viral gene. So injected its DNA, and it's going to make these things. What is the nucleotide? When we say the host provides the materials for making the viral components. So if the material is nucleotides, what's the viral component that this is making? We have another question? Yeah. All right, let's answer this one first. So the nucleotides, what is it going to be used to make? Yeah, so the nucleotides are used to make the nucleic acids, right? What are the amino acids going to be used to make? The viral proteins, right? And so therefore and then the energy, ATP is used for energy to do that. And so the host cell is providing all of this for the virus. Now you have a question.
So now we talked about how bacteria take in DNA from the surroundings, like our cells home. So is there certain viruses that just spew out their DNA that doesn't area and just bacteria can take it up and then they'll start replicating the virus?
Yeah, that's a good idea. No. No, it doesn't. They actually do infect it, but you're right that they do that. And we're going to talk a little bit later about some mechanisms that the bacteria have that destroy that type of DNA that it takes up. All right. Okay, so let's look at this picture here and talk about this. This picture shows you in picture format what we just wrote down. So at the top here, you have your virus. It has the capsid proteins, and this is a double-stranded DNA virus. And so the double-stranded DNA is its genetic material. So here it's showing you the virus is going to infect its host cell here. With this arrow going in here, what is it not showing you there that we've talked about? What has to happen for that to occur? Nathan? Yeah, it has to bind to a receptor on the cell. So remember they have to have the right quote-unquote fit. And so there's a receptor here. So you have to have the right receptor on the host cell in order for that virus to bind. If that receptor is not there, then that virus can't infect that particular cell. So it has the receptor, and then it enters into the host cell. Notice that there's two pathways here. So this way here, what are these little purple dots here? What do you think they're trying to show you here in this picture? So from here to here, what are they trying to show you? The protein coat or capsid breaks down, okay? So, very good. So the capsid breaks down. What that does is it exposes, I'm going to put here, exposes the viral DNA. So the viral DNA now is hanging out in the host cell. Then remember, the host cell is going to be used to make new viruses. So what are the two main components of viruses? Proteins and nucleic acids, like their genetic material. So therefore, notice that there's two arrows here. 1, 2. What are we going to do? The first one is we're going to use that viral DNA. So let's say this is a human cell, and it now has this viral DNA in it. Our cells can replicate our own DNA, but when virus eject their DNA into us, we can also replicate their DNA using our enzymes. So therefore, that's what this is, it says replication here. You're making many copies of viral DNA. So we're making, I'm just going to put making copies of the viral DNA. So that's what they're showing you here. Using DNA polymerase and DNA ligase and all of the enzymes that we use to replicate our DNA. Now we're just replicating the virus's DNA, which is what the virus wants us to do. Then we can also take the viral DNA, and what's these two steps here? What is collectively this was going on here? Transcription, translation, whole thing.
Protein synthesis.
Yeah, protein synthesis. So right here to right here is protein synthesis. We're going to use the viral DNA to make viral proteins. So it says here, this is transcription right here, from DNA to RNA. Then from here to here, this is translation, making the capsid proteins. The whole thing is protein synthesis. So we're making the proteins. So now we've manufactured both parts of the virus. And so now what this picture here is showing you is the assembly, self-assembly of the new viruses. So the capsid proteins are going to surround the genetic material.
Percy.
Hello. And then exit from the cell. I have him in the tower. And they exit from the cell. And so what do you think they're going to do when they exit from the cell? They're going to go infect more cells. And notice there that this is replication, because you started out with one virus and now we end up with many viruses after this process. So they go infect more cells. Yes? It depends on the type of, because this is a generic idea here. Sometimes we're going to learn that some viruses take their viral DNA and insert it into our DNA, which can affect the way we transcribe proteins. Sometimes the viral DNA, some of the proteins we have some of the, when it use the genes on the viral DNA can make proteins that we have adverse reactions to. Sometimes it actually damages the cell itself. Sometimes it kills the cell. So it depends on the virus, on the type of virus, and so that's why we can have adverse reactions to, different viruses. Each reaction is kind of different.
This one? like a lot of what we call getting sick, like our body fighting the virus and not what the virus does to us, but what we try to do to the virus? Like
Yes,
patches and
absolutely.
stuff like that,
Yes.
fever.
Yes, yes, absolutely. So those things are good, it just makes us, yes, absolutely. And I touch on it a little bit later on, but absolutely. Okay, so this is the general overview of how a virus works. So what we're going to look at next in looking at viruses, we're going to look at viruses that infect bacteria. And so they don't have anything to do with us, but the reason why we need to look at that is because there's some things with the viruses that infect bacteria that we use in DNA technology. And then we're going to look at viruses that infect us and some things about them. All right, so the bacteriophages, the viruses that infect bacteria. So these guys reproduce in a couple of different ways using the Lytic or Lysogenic cycle. The Lytic cycle results in the death of the host cell. So this is for bacteria. And so it's going to kill the bacteria that it infects. And so in the last stage of the cycle, the infected cell lyses or breaks open and releases the virus produced within the cell. So kind of the big picture of what happens with the Lytic cycle is if you have a bacteria and it comes and is affected by a virus, it injects the genetic material in it and basically causes this bacteria to become a virus-producing factory. So it begins to replicate the viral DNA, make more viral proteins, and basically inside of this bacteria cell, it's going to become so full of viruses that it's actually going to break open and all the little viruses can go out and infect new cells. So it kills that host cell, but in the meantime, by killing that one host cell, it gains a whole bunch of new viruses that can go and infect different cells. And so that's what it means here by cell lysis. I used this word when we did osmosis, when water goes into the cell and it swells up and eventually bursts. Each virus can spread to infect another healthy cell after that. And so that's what this picture here is, the Lytic cycle. And so at the top here you have your bacteriophage. And notice it says here attachment. So why does it attach to this one? Remember it has the host has receptors. Remember that it attaches to. And so therefore that bacteriophage is not going to affect just any bacteria. There's thousands and thousands and thousands of different species of bacteria. So it only affects certain ones and the ones with the right receptor. And here, inside the bacteria, what's this line? It's its DNA. It has one, remember, big circular chromosome. So from here to here, what is happening, and it says here what's going on here, the virus is injecting its DNA. So let's say this is a DNA virus, and so it's enterophage DNA, that's this part here. And then if you notice here, the bacteria DNA itself is all broken up. So it's all broken down here. And the virus causes that to happen. So the degradation of the host DNA is right here. So those two things occur. Why is it beneficial for the virus to break up the host cell's DNA? So it injects its DNA, but why is that? Why is breaking this DNA up important? How is it helpful? Lucas?
Is it like prevent the cell from making its own DNA again, so it doesn't waste like stuff that the virus wants to use?
Okay, yeah, so it's not going through protein synthesis and using its own DNA, and so therefore it will only use the viral DNA. All right, and so therefore all the resources and stuff like that will be used to go to make copies of the viral DNA and make new proteins to make new viruses. And so that's exactly right. So what from this picture to this picture, it's starting to make more copies of DNA and more proteins. And then notice here, assembly, what's the assembly of? They're assembling the viruses. The assembly of the the viruses inside the cell. And so all the different proteins come together to form your capsid surrounded by your DNA. And so you make lots of different viruses, and the cell lyses, so this is the cell lyses, explodes these bacteria. Now, bacteriophages can go and infect different cells, and the whole thing starts all over again. So then one of these goes and infects another cell, and so on and so forth. And so that's how the viruses reproduce, as through the lytic cycle. But the lytic cycle kills the host cell every time. So what we're going to look at is a lot of viruses in conjunction with the lytic cycle go through another thing called the lysogenic cycle. Yeah, Liam.
I was going to ask, so for the lysic cycle, would it be possible for humans at least to have, if you're infected with a bacteria and you then inject like a bunch of viruses that are bacteriophages that are hunting that specific pathogenic bacteria, and they just, because they only bind to that specific bacteria, would it be possible like for that as a treatment?
That could be, yeah, that could be. Absolutely, as long as you're 100% sure you're not going to be affected by it. Yeah, absolutely. Yeah, it's a good idea. The lysogenic cycle does not destroy the host cell, and instead of replicating the virus itself, the genome replicates. So let's look at that. So this bacteriophage called the lambdaphage infects E. coli, and it does both the lytic and the lysogenic cycle. So when we're looking at this, infection occurs just like I just showed you, where it lands and injects its DNA. But what happens next depends upon if it's going to go directly into the lytic cycle or the lysogenic cycle. So let's look at this new part here, the lysogenic cycle. During this cycle, the viral DNA molecule is incorporated into the host cell's genome. This is different than the lytic cycle. So if this is your bacteria, and this is the bacterial's chromosome, when this bacteria is infected by the bacteriophage and its genetic material gets put into the cell, instead of this genetic material being used right away to make viral proteins and viral DNA and make new viruses and the bacteria explodes, instead of that, what happens is the virus puts its DNA, it actually cuts open the bacteria's chromosome and inserts its DNA, so that the viral DNA is actually part of the bacteria's chromosome now. Why is this beneficial for the virus? See, when it does this, it hasn't reproduced. You don't have more viruses right now. You just have a bacteria now with viral DNA. Why would that be beneficial?
Because the infected cell can be used over and over to make viruses.
That's right. This infected cell can be used over and over again to make new viruses because it has the genetic material. And when we think about being used over and over again, realistically, bacteria cells, they reproduce like crazy. They divide. You can get 1 million bacteria cells in a day. All right, they're they divide very rapidly. So what happens is when this bacteria cell divides, what does it do as part of cell division? It makes a copy of its DNA, right? Well, guess what's in its DNA now? The viral DNA as well. So now you have 2 bacteria that contain the viral DNA. What are those bacteria going to do? Divide, you'll have 4 bacteria, and you get it, and so on and so forth. So still now let's say you have 1 million bacteria that have this viral DNA. At any given point, this viral DNA actually pops out of the chromosome. So it actually becomes not part of the chromosome anymore. And that viral DNA is used to make more DNA and to make proteins, which come together, and new viruses can be released and be used to go infect other cells. So the benefit of having the DNA become part of the bacteria's chromosome is that they're using the bacteria and allowing the bacteria to divide, and now you've got a whole bunch of bacteria that have the viral DNA, And then all of these bacteria can eventually make new viruses from that.
So the DNA that pops out of the host cell's DNA, does it then go through transcription, all the way to RNA and protein?
Yes, yes, absolutely, absolutely. And so, so oftentimes, some viruses will do this where the DNA lays dormant for a while in the organism, and then it pops out, and then when it does that, it enters the Lysogenic cycle, and when it does that, when these viruses are released, it will lyse the cell, and the cell will die, but that's okay because you've got almost 1 million other cells that also have the virus DNA inside of it, okay? So when the host cell divides, it copies the phage DNA and passes the copies to its daughter cells. And so therefore these viruses propagate or kind of divide and make copies of their DNA without killing the host cell. And as I said, then they can trigger and go enter the Lysogenic cycle at a later time. So let's look at this picture here, because this shows you the comparison between these two cycles. So on the left is the one we just looked at before the lysogenic. So the bacteriophage is injecting its DNA. There's its DNA. The DNA is used right away to make new viral proteins and capsid proteins, the capsid proteins and the genetic material, and the cell lyses. And so therefore this, the Lysogenic cycle, we could say reproduces the virus. Because you've gone from one to now the cell lyses, and you've got many that can go and infect other cells. But let's look at the lysogenic cycle. So the lysogenic cycle starts here. So this is step 1 for both of the cycles, where the phage is injecting its DNA into the host cell. This is step 2 for both of the cycles, where now here's the DNA. But step 3 for the Lysogenic cycle was reproducing viruses in the cell lysing. So that's what happened there. For the lysogenic cycle, step 3 is instead of going and being used to make new viruses right away, that is viral DNA is injected into the bacteria's chromosome. So it says here, it says phage DNA integrates into the bacterial chromosome, becoming a prophage. So prophage is the viral DNA that has become part of the bacteria's chromosome. So I color-coded this because that's different here. So this part right here, this is prophage, which is this, what we just wrote here. So what does that bacteria do? It divides, and when it does, it replicates its DNA and therefore replicates the viral DNA. And many cell divisions, they're only showing you one here, many cell divisions produce a large population of bacteria. So you can get 1 million bacteria now that are millions that now have this viral DNA that are infected with this prophage. So let's, out of those millions of bacteria, let's say 1 here, what happens is usually what triggers for the DNA to pop out here is some kind of environmental stimulus. So an environmental stimulus triggers the viral DNA to, and I'm going to put quotes here, to pop out of the bacteria's chromosome. And enter the Lysogenic cycle. So we have here popping out here. So this was step 4, step 5, all right? So notice here, at the end of the Lysogenic cycle, we're back to where we started out with. It pops out, and now, when it pops out, it can now enter the Lysogenic cycle and go in this way. So that would be 7, 8, and 9. All right, does that make sense? Okay. And so then it starts using that DNA and enters. So I'll just do 7, 8, and 9. All right. So So we can go around this way, or you can go around this way and go back to that, all right? And so therefore, the lysogenic cycle, the lytic cycle reproduces the virus. What would we say about the lysogenic cycle? What does it reproduce? What does it reproduce? The viral DNA within the bacteria cells. So this reproduces viral DNA using the bacteria to do that. So that viral DNA is within the bacteria. Okay. And so those are viruses that infect bacteria. So so this is not our cells. And so bacteria are unique in that they're unicellular. And so when we look at a colony of bacteria, let's say here's a bunch of bacteria here. So we're talking about this one single virus can infect a colony of bacteria very, very quickly because if you have this bacteriophage that comes and injects its genetic material inside the cell, and let's say it enters the lytic cycle. What does that mean? It's going to use that genetic material right away, make new viruses, and so it's going to kill this cell, but in the process of killing that cell, it's going to release, I'll just put V's for viruses, new viruses. And those new viruses can now go and infect these cells and so on. So one virus can wipe out an entire bacteria colony very quickly because the virus only has to infect the one cell because bacteria are only one cell big. The lysogenic cycle is helpful because if a bacteriophage infects this cell, and instead of entering the lytic cycle, it enters the lysogenic cycle. Does it kill this bacteria? No. So it has the genetic material in there, and so while this virus, all the ones may kill off all of these guys going through the lytic cycle, this one that's going through the lysogenic cycle, what it'll do is allow this bacteria to divide, and now you'd have two bacteria with the viral DNA, and so on and so forth. So it's helpful because it's not killing off its host, but yet still replicating the DNA. So both of those in conjunction with each other are helpful for the survival of the bacteria. All right, works better here. So think about us when we get infected by a virus. We have a whole system, the whole immune system, to fight off infections. Little bacteria cell, I mean, they're only one cell big. They don't even have tissues. They're one cell big. So, but they do inside their cell have some defenses against infection. So, a phage, as I said here, can wipe out an entire bacterial colony very quickly. So, natural selection favors bacterial mutants with receptor sites that are not recognized by the phages. Remember, mutations are random. They're changes in the DNA. So it can be caused by external factors like ultraviolet radiation, different chemicals in the environment. So if a bacteria randomly mutates, so let's say this bacteria here mutates, and let's say the mutation occurring in this bacteria, the mutation is in the DNA that makes the receptor sites for the phage to bind to. Hi.
Hi, can I see Abby Horning and Kate Rogowski?
Sure. So let's enlarge this. Let's say these viruses right here that gets infected, they get infected by the bacteriophage because they have specific receptor proteins in them. And so the virus then can come and land and bind to these receptor proteins and inject its DNA and affect it. But if you have a cell with a mutation, so let's say the mutation affects the gene that makes those receptor proteins. So let's say they don't have the receptor proteins, or the receptor proteins aren't the right shape, Is the bacteriophage going to be able to infect it? No. And so therefore this would be a good mutation for a bacteria to have. Now bacteria cells can't go, ooh, we need to mutate because there's a virus infecting us. Then that's not what happens. The mutation is random. So the bacteria is just living fine, and one little bacteria mutates, and right now, before the virus infects, the mutation doesn't really harm it maybe, or doesn't really help it either. But then when the environment changes, and now you have this bacteriophage in their environment infecting this colony of bacteria, now that mutation for that bacteria is a good mutation because it's going to not be infected by that virus. And so therefore that mutation is going to spread pretty quickly in the population. How? Because this little guy right here is not going to be infected by the virus. What is it going to do? It's going to replicate, and millions, and in 24 hours we can get a million cells in the bacteria that virus can't infect anymore. All right, and so this is why bacteria. Have you heard that bacteria mutate very rapidly? This is why, because they're single-celled and they reproduce very quickly. This is way different than us. Think about us and how we would have to change the genetics of a population. So if I had a mutation, we're billions of cells. If I had a mutation that occurred in my skin cell, is that going to affect the rest of the population at all? No, it's only going to infect me. So how can I get a mutation that would affect the whole population? What cells in my body or your body would have to be mutated? Your germ cells, the cells that are making your egg, your egg and your sperm cells have the mutation, because those are the cells. If a mutation occurs in your egg and your sperm cell, you have a child, you pass that mutation on to your child. That child has to grow up and then have kids and pass that mutation through their egg or sperm cells to the next kid. And so for organisms like us, for a new mutation to spread, it takes a long time because it has to occur in a certain cell in our body. We have to pass it on to our kids have to grow up, they have to have kids, and there's a lot of generation time. So it happens, that's how we get like big R's and little r's for genetics, is because of mutations, but it takes a while for it to spread throughout the population. Not so much for bacteria, because bacteria is only one cell big. That mutation happens in that one cell. It affects the whole organism. They divide. Now you've got two organisms that have it, and so on, and you get mutations very, very quickly as a result of that. That is great for the survival of bacteria. That's not so great for when we have infections that are caused by bacteria and we're trying to combat them. The fact that bacteria mutate very quickly is not good for us, but it's great for the survival of the bacteria.
So viruses mutate quickly too?
So yes, and viruses mutate quickly as well. For the same type of reason, because they're using the bacteria, they can use cells and so on to divide and so on. So yes, and that is why we need a flu shot every year. Because viruses also mutate very quickly, and so what they do is they take, the top. It used to be just 3. This year they came out with a quad flu virus that had 4 different strains in it. For the top 4 from last year, and they're taking a guess which ones are going to be spreading throughout the human population this year, but based upon what happened last year. But viruses mutate, and so therefore, if the virus mutates, and it gets into the population, then you've got a bunch of people getting the flu, even though they had the flu shot, because the virus mutated. And there's no way that somebody could predict that or make that flu shot, better because of that, because you can't predict the random mutations that's going to occur and so on. So some people say the flu shot doesn't work or the vaccine doesn't work. That necessarily isn't technically true, because it works against the viruses that it's made for. It's just that you can't predict. It's hard to predict what viruses will be around, all right, in the next year. All right, time to get out of here. Now, okay. You guys are so good. You didn't